Assalamualaikum warahmatullahi wabarakatuh and a very good day. My name is Dr. Muhammad Azrin bin Ahmad. I am a senior lecturer at Center for Mathematical Sciences, University Malaysia Pahang. In this video, I would like to walk you through our invention entitled Biological Perspective of Nth Order Limit Language. Co-inventors of this invention are Siti Hajar Muhammad Khairuddin, a Master by Research student, Associate Professor Dr. Aizi Nurmazila binti Ramli from Faculty of Industrial Sciences and Technology, who is the person that advised us regarding biological information, and last but not least, Dr. Nur Azia binti Azhar. Before we go further, allow me to introduce splicing system and splicing language. Splicing system is a study that focuses on the recombinant behavior of double-stranded DNA when acted on by restriction enzymes and ligase. Meanwhile, splicing language is a product resulted in splicing system. There are a few types of splicing language, but our result focus on an extension of limit language, which is nth order limit language. The sole reason we include biological elements in our research is to verify the result we obtain in our theoretical works. We have improved the definition of nth order limit language, where right now we focus on the number of rules used in the splicing system. The rule used in the splicing system must possess certain requirements. Later, you will see that the number of rules used in the splicing system affect the order of limit language. In this experiment, due to cost and time constraint, we propose these two models that use 3 and 4 restriction enzymes respectively. By looking at this video, the solution that contains DNA strands will be mixed with restriction enzymes and ligase to complete the digestion and ligation process. The solution is then observed by using the right equipment. Here are the equipments used in this experiment. Bio-red cycler thermal cycler is used for polymerase chain reaction PCR process, where we generate 1,000 copies of initial strand double-stranded DNA. It is also used to keep solution which has been aliquoted into test tube under certain condition. The solution we obtain after digestion and ligation are aliquoted into test tube and keep under certain condition to observe the process. Gel electrophoresis is performed where the aliquots are put into wells in the gel to observe the digestion and ligation results. The gel is then visualized using blue LED transilluminator under UV light to observe the digestion and ligation results. So, this is the operational framework of this experiment. We start by developing the mathematical model to be used in the wet lab experiment, and by using the right procedures, we conduct the experiments. If the experiment is success, we further analyze the experimental results and reach the conclusion. Now, let me explain the result we get from the experiment based on Model 1. Model 1 uses 3 restriction enzymes, and the splicing system is modeled as follows, where we have a set of alphabet A, a set of initial string I, and a set of rules B. Meanwhile, we denote C as an empty set. This is the third order limit language according to the model we use, and the string is divided into these few fragments, and the length of each fragments are given as mentioned on table 6. Before we run the experiment, this is what we have predicted according to the length which have been tabulated in the previous table, and right after we visualize the gel under UV transilluminator, we will obtain this image which somehow mimics the one we have predicted before we run the experiment. This experiment will be helpful to the research that study DNA splicing system, where the splicing language, namely nth-order limit language, has been proven theoretically and biologically in this wet lab experiment. We have published 5 papers related to this project, where 4 of them are published in Scopus Index Journal, and one of the paper is published in non-index journal. We have also filed a copyright regarding the experiment to Intellectual Property Corporation of Malaysia, my IPO, on 18th August 2021. I would like to thank Research and Innovation Department, University Malaysia Pahang for this opportunity. Stay safe and see you around. Bye!